We will very briefly discuss mitosis, a process to help create new somatic or body cells. The cell cycle begins with the interphase stage, where the cell experiences growth and DNA is replicated. Chromatin, or long strings of DNA, condenses as enzymes coil them to form chromatids. The copies of the DNA join together at a centromere to form sister chromatids. Then, mitosis begins. Prophase takes place, where the sister chromatids coil and move together to form chromosomes. Metaphase is next, and the spindle fibers line the chromosomes by their homologous pairs, chromosomes that contain the same alleles. In the next stage, anaphase, spindle fibers attach to one of the homologous pairs of chromosomes and pull them to opposite sides. During telophase, the cell lengthens and new nuclei are starting to form around the chromosomes. Finally, the cell splits during a process called cytokinesis, forming two new daughter cells. These cells are classified as diploid, as they have two copies of each chromosome, one from each parent. They are completely identical to the parent cell.
Before the first phase of meiosis begins, DNA replicates, and the two copies bond together through the centromere. The DNA is replicated so that the new cells and offspring can have the correct instructions. Every cell needs DNA, and replicating it allows the parent cell to keep all of the information while the daughter cells can possess the same information or just a piece of it. Enzymes begin to coil the DNA together as they wrap and tighten to form chromatids. They are called sister chromatids, and together they form a chromosome. Homologous pairs, or chromosomes that contain the same genes, move together. Prophase 1. In this stage, chromosomes become shorter and thicker, and the homologous pairs join together through a process called synapsis. This creates a pair of sister chromatids and forms a shape called a tetrad. Next, the four chromatids have alleles that are broken out of the DNA and recombined on a different strand. This process is called crossing over, and new gene combinations are formed between the chromosomes. These gene combinations are random and are also referred to as independent assortment. The result of crossing over is genetic recombination in the cell. Prophase 1 is the longest phase of meiosis, accounting for 90% of the time spent.
Metaphase 1. The sister chromatid tetrads line up in the middle of the cell. Spindle fibers are released from opposite ends of the cell structures called centrioles. They attach to the centromeres of the tetrads. Anaphase 1. In early anaphase 1, the spindle fibers pull apart the homologous pairs as they move back to their designated centrioles, dragging one pair of the sister chromatids to each side of the cell. The tetrad of four chromatids now is broken up into pairs. In late anaphase 1, when the sister chromatids have almost returned to their microtubules, the cell membrane begins to restrict. Telophase 1. The two sets of sister chromatids are each enclosed by a nuclear membrane as the cells begin to split. Cytokinesis occurs and produces two cells, each with the same number of chromosomes as the parent cell. However, these cells are not identical to the parent cell. Not only has genetic recombination had an effect, but each cell consists of sister chromatids from one parent instead of a homologous pair from both. They are already haploid cells, despite having the same number of chromosomes as a diploid cell. This is another way in which meiosis differs from mitosis.
In prophase 2, the chromosomes do not duplicate. All the DNA is already present. However, centrioles duplicate so that spindle fibers can eventually latch onto each chromosome. In metaphase 2, the chromatids line up in the middle of the cell and new spindle fibers form. In anaphase 2, the spindle fibers latch onto the centromeres of the sister chromatids and pull them to the opposite sides of the cell. They are now single chromosomes. In telophase 2, four daughter cells begin to form, and cytokinesis occurs, resulting in four new gamete cells or reproductive cells. Each daughter cell has exactly half the number of chromosomes that the parent cell had and has one copy of each chromosome.
Finally, we will talk to you about fertilization. Fertilization occurs when a sperm cell and an egg cell bind together to form a diploid cell. Although many sperm can bind with an egg, only one can fuse with an egg plasma membrane, injecting its nucleus and other organelles into the egg cytoplasm. If more than one sperm fuses, it results in polyspermy, which ends the cell's life because it obtains too many chromosomes. To stop polyspermy from happening, there are two defense mechanisms. One is a rapid depolarization of the egg plasma membrane, caused by the fusion of the first sperm. This depolarization prevents more sperms from fusing with the egg. However, this only acts as a quick prevention, for soon after fertilization, the egg returns to normal. However, then the second defense mechanism comes into play. The second defense mechanism is the egg cortical reaction. After a sperm fuses with the egg, there is a release of calcium. The calcium changes the structure of the zona, hardening it and inhibiting sperms from binding with it. The zona is the structure around the egg that the sperm binds to. Zonas make sure that more than one species can't cross fertilize.